My name's Naomi Wray. I'm from the University of Queensland in Brisbane in Australia. I'm going to start a little bit about myself. So I'm probably known now for working on genetics of psychiatric disorders. But I feel like the thing that I bring to the table is a training in quantitative genetics, and so applying quantitative genetics theory to understanding the results we get. So, for example, this paper, which is a review paper published this year, is a kind of capstone of some of my work from the last 10 years. But today I'm going to talk about DNA methylation, which I feel like is a bit like bringing coals to Newcastle, because there's a lot of people here who know a lot about analysis of DNA methylation data. But one of the reasons I chose this is because at the time I was asked for a title. It was something I was working with, and I have been working on DNA methylation data over many years, and the work kind of illustrates the way that our group operates. So Peter, who talked the other day, and myself and Zhan Yang run the program in complex trait genomics. That's our group. We're all dry lab people, and the work that I'm talking about has really come about because, having worked on DNA methylation data, I decided I wanted to generate some DNA methylation data in a case control setting. And having worked on DNA methylation data, Zhan Yang decided that he'd like to try a new method and develop a software. And so these two things were going on at the same time. And this work really kind of illustrates how method development takes place and how the analysis of the data helped to integrate with the method development. So the other thing I want to tell you about myself is that my first degree was in agricultural science. My first paper in 1986, which was many years before many of you were born, was on analysis of gestation length in beef cattle, which is a very difficult trait to measure because you have to know when the cow is mated and when the calf is born. But still, 30 years ago, I was working on a data set of over 75,000 records with a very complex genetic structure. I was estimating the direct genetic effect because of the influence of the calf on when it was born and the maternal genetic effect. So I was working on a genetic model which still today in human data, we would find it difficult to generate the data sets to look at that. In livestock, we have many longitudinal records. So what I'm trying to get at is there's many analysis methods applied in livestock which are actually really relevant to human genetics today. And that's part of what I think Peter and I have brought to the human genetics field, is this understanding from livestock genetics. Many people in our group come from agricultural genetics. Zhan Yang, his PhD was on epistasis in rice. So this is a very nice segue into my conference, which you've already heard about, but I won't tell you about it again. So the conference on quantitative genetics, what's unique about it is it brings together people from across different fields, from evolution, from model species, humans, livestock, crops, methodology, and it brings us together because of the interest in methodology. So it's a conference we're hoping for about 600 people. It's going to be very special. There's some posters out there that you can take home, and also some bookmarks which you can take away, and it shows nice things like koalas and blue seas, because even though it's winter, it's actually really nice there. In fact, this is a bit of a cold day for our winter. Okay, so I'm talking about DNA methylation. So what do you need to know about DNA methylation? That in the genome, you often get a C followed by a G. They're linked by a phosphate bond, so we call those CPG sites. And often those sites can attract a methylation, a methyl group. So often you get these CPG islands near in promoter regions of genes. And in the unmethylated state, the gene is it's associated with expression of the gene. And in the methylated state, suppression of the methylation. So that's kind of all you need to know. So we can measure DNA methylation through sequencing, but we can also measure it in array technology. And so down the bottom there, I've got the Illumina array for SNP typing and the Illumina array for DNA methylation typing. And you can see very similar. They use the same kit for scanning. You just need to do a bisulfite conversion pre-preparation in order to detect the methyl group. The only thing is you do need 5 times more DNA for the DNA methylation, but other than that, it's very similar technology. So in my talk, I'm going to refer to the Lothian birth cohort data. So the Lothian birth cohorts are have been collected by Ian Deary, who's our collaborator at the University of Edinburgh. The Lothian birth cohort people, their birth cohorts are born in 1921 and 1936. The sample size is about 1,300, and they have Illumina 450K methylation data and also directly measured blood cell types. So many of you are familiar with the genome-wide association study where we test for the association between a trait and a single nucleotide polymorphism, or we can conduct a DNA methylation study in the same way, association study in the same way. Now we're trying to detect the association with a DNA methylation probe with a trait. So very similar in concept. But I did a PubMed search last week, trying to find the number of papers which have been published for genome-wide association studies compared to methylation-wide association studies. I might not have got my search terms exactly right. There's a lot of hyphens going on around the place. But I think this ratio is probably pretty realistic. 17,000 for genome-wide association studies, only 400 for methylation-wide association studies. So we can ask ourselves, why are there so few methylation-wide association studies? Anyone want to make some suggestions?
Very expensive.
Expensive, yeah. And why are they expensive? Well, I'm not talking, so I work on variation between people, so that means we want large samples, which means we tend to use array technology. But the arrays are about 8 times more expensive than the genotyping arrays, even though it's the same technology. And you can ask yourself, why is that? And it's really because there's no competition. There's no real reason other than that. Any other reasons? Yeah.
Hard to correct for cell type composition?
Hard to correct for cell type composition, yeah, so there's confounders. And so with the genome-wide association study, our kind of underlying hypothesis is one of causality, whereas with DNA methylation, there could be confounders, and also we kind of stepped away from that causality because what we measure may be a reflection of maybe reverse causation, maybe consequence as well as cause of the trait. There's one other reason, and I think I've sat on many grant review panels, and I think that as soon as you say you're doing an epigenetic study, it will go to the epigenetic panels, and in the old days at least the epigenetic experts were mouse epigeneticists, and they would say things like, well, what's the point of measuring DNA methylation in blood? That's not the tissue of interest for the trait you're interested in, particularly when I work on disorders of the brain. And they'd also say DNA methylation is you're measuring these epigenetic marks on an array, you're only looking at CPG sites, it's only one of few epigenetic marks, it's not very useful for biology. On the other hand, I would often also then sit on mental health panels or neurology panels, and there you get multitudes of people who are interested in biomarkers for diagnosis, and all the criticisms, I think, which can be put at DNA methylation can be put at all those biomarker studies, but those are all candidates. And I think we spend a lot of money on biomarkers, and I think DNA methylation may be a more interesting way to think about biomarkers. So there's a couple of papers. One is evaluating the Illumina latest array, the EPIC array, and its coverage, and the other one is talking about some of the issues to do with DNA methylation studies using the particular problems associated with case control studies that I'm going to talk about. So for people who aren't familiar with DNA methylation, just a few things to think about. So say I'm thinking about taking DNA methylation from blood because in large samples that's where we have our samples available. And of course, how do we measure DNA methylation? Well, we're taking the buffy coat from the blood sample, white blood cells, so we know that our sample is a mixture of blood cell types. And the methylation, there's different methylation signatures for different cell types, and so what we might be detecting is something to do with cell type composition, as suggested here. And so there is a way around that. We can take samples which have been previously where we have cell types measured, and then we're able to make predictors of cell type composition. And that's quite well established in the field. This is the application to our Lothian birth cohort data where we have the cell types measured, and you can see that the pattern of proportions of cell types is very similar from the measured versus the predicted. And at the top I've got the correlations between the cell types from the measured values and then the measured versus predicted. Another thing is that DNA methylation changes with age. And so particularly in case control studies, we might be concerned about differences in age confounded with our case control status. But again, with samples where age has been known and measured, there are now reference samples, reference predictors, which allow us to directly predict a person's age from their DNA methylation data. So there's this very famous paper from Steve Horwath, who I think is from around here. And this is showing, I didn't apply it to the Lothian birth cohorts in this example because they're birth cohorts, so it's not very helpful for showing the age predictor. But you can see on the x-axis the actual recorded age, and then the y-axis the predicted age. And you can see, and these were actually in Chinese samples, so showing this predictor actually goes across ancestries as well. So of course that age predictor, it's a predictor, which means it's predicted with error. So one of the papers that Peter's PhD student has worked on has been using larger sample sizes to develop the age predictor. And what we're finding is that now the difference between the predictor and the actual age is much smaller. And you can now think for yourself what that means about some of the work about DNA age acceleration, which is where people have looked at the difference between the predicted and actual age. Another thing, well, maybe one of the reasons we're interested in epigenetic marks is it's a way that the environment can impact on biology. And so this is an example, one of the first things that was identified through DNA methylation studies was detecting associations with smoking. And so what I've highlighted on the y-axis is just the size of those minus log 10 p-values, 180, and the bottom one was 60. And this was a study with a discovery sample and a replication, showing that you're getting very similar signals. And so these are real associations. And the other thing to take away is actually the sample size. If you're used to the sample size that we need for genome-wide association studies, these sample sizes are really very much smaller for identifying these really massive effects. So one of the first studies we were involved in was, again, with the Lothian birth cohort data. And this was, here we looked at two traits, height and BMI. And here we've got the QQ plot, so we're comparing the minus log 10 p-value on the x-axis with the observed minus log 10 p-value. And under the null hypothesis, we expect the associations to follow the red line. And you can see with height that there's no evidence for any associations of DNA methylation with height. Now that maybe makes sense. These are older people. Why would there be methylation in height at that, associated with height at that age? Compare that with BMI, and you can see that the black dots really raise very sharply above the red line, a massive inflation. So if we only had the right-hand side data, we might have not gone ahead and published it because it just looks so massively inflated. On the other hand, we can argue, based on height, there's no evidence there's anything wrong with our data. It's just that it's something about the DNA methylations associated with BMI. There's just many of them floating around in blood. It may be a reflection of differences in cell type proportions. You can imagine that BMI is also a reflection of what people eat, so maybe that's also why there's many things that you can pick up floating around in blood. So one of the key statistics that we might think of is the genomic inflation factor. So that's the ratio of the observed median test statistic. So the median would be about here compared to the expected. Here you see no genomic inflation. Here you see a massive amount of genomic inflation, raising the median, raising very much above the line of expectation. So one thing that we did was that we took these data into a prediction scenario. So now we've got the data from the Lothian birth cohort and another sample, the Lifelines cohort. And we made, just like we can make polygenic risk score predictions with DNA data, which we've done here, which is the green bars, we also made methylation predictors, which are the red bars. And not surprisingly, from the QQ plot on the previous slide, the methylation prediction score had no effect in height, whereas we do have good prediction for the GWAS polygenic risk predictors. But for BMI, you can see that the red bars are actually very high. So there's good predictive ability for BMI. And so the blue bars next to the green are showing both the methylation and the GWAS polygenic risk predictor together. And you can see they're actually additive, they're not multiplicative. So we were getting additional prediction from the methylation over and above just genetic prediction. And again, one of the take-home messages is that for the genetic polygenic risk predictors, these were based on these massive discovery sample sizes, 350,000 for height, 250,000 for BMI, whereas very much smaller discovery samples for the DNA methylation. So it was these results that made me interested in generating some DNA methylation data for a case control cohort, thinking that this might be useful in a prediction and to aid diagnosis. And just to reiterate that in disorders of the brain, there's a very big push and a need for better diagnosis and looking for biological help in prediction. So this is a basic model of analysis for DNA data. So we have our phenotype Y, a linear model with some covariates, and then we want to estimate the effect of each probe, taking each probe in turn. So this is a basic model of analysis, and really it's been shown many, many times that this model is not good enough for DNA methylation data because there is some massive correlations across the genome. So how can we improve on that model? Well, we can fit known covariates, for example, cell type proportions, even if they're predicted cell type proportions, and known covariates such as smoking. We can also take the DNA data, the DNA methylation data, and generate principal components, fit those principal components much the same way as we do in genetic data. But again, those basic corrections have also found to generate massive inflation. So now there's many methods available for analysis of DNA methylation data. And I've put some of them up here. The surrogate variable analysis goes back to 2007, used for all sorts of omics data and is really quite robust and stood the test of time. A paper here in 2014, this is the E-washer method. This is actually a linear mixed model which fits the variance-covariance matrix from the DNA methylation data, but it doesn't use all probes when it makes that DNA methylation variance-covariance matrix. A method from the group here, refactor, which is a principal component method, again thinking about this, the problem with cell type heterogeneity. And another paper published this year, the latent factor mixed model. So I'm just highlighting those papers because in our new method, we've been comparing to those methods. So in the new method that we've brought in, we call it MOA, Mixed Linear Model-Based Omic Association. In many ways very similar to the E-washer method and very similar in concept to the GREML method that's used in genetic analyses. So rather than, you can see we've got a mixed model, here we've got a random effect, which is the genome-wide DNA methylation value of an individual, the variance-covariance matrix. You can see we're building a covariance. The value between any pair of individuals is essentially the covariance between all their DNA values genome-wide. And then really this was the method that we were introducing, and then as a consequence of the simulations and analysis, we brought in a second method which we called MOMENT, which essentially adds a second random effect where we're splitting the DNA methylation probes based on a criteria to better account for some of the things that we see in DNA methylation data. So this method is, as I say, it's very much led by my colleague Zhan Yang and Futao Zhang, that was the postdoc who did the work. And it was just published last month, I think, in Genome Biology. And so this is called OSCA, for omics-based complex trait analysis. So because this is a methods-orientated course, I thought what's interesting is to think about the way in which we approach developing a new method. And so you'll see in a paper the way that we develop a new method is we think of the theory, we develop the method, we then apply it to simulated data where we look at different simulation scenarios, change our parameters, and then we apply it to real data. Just how many people here have actually used simulation in their work? Okay, some, half. Good, well, I'm glad it's only half because I'm going to talk about the way in which we use simulation to help develop methods. And so one thing when you read a paper or you're where it's using simulation and when you're doing this yourself, the key thing that you have to think about is when you set up your simulation, are you biasing your comparison to favour your method? Because often the way in which you think about simulating is very much aligns with the way in which you've developed your method. And so that's sometimes why you see new methods always do better than previous methods. Of course, when you develop a new method, you've got no guarantee it's actually better than the previous ones, and quite often you can do a lot of work and then you get to the point where you think, actually, this isn't contributing anything more than what's already out there, and then you have to start again. So in a simulation, you have to think, how do you simulate and how do you evaluate? So in this case, So one of the issues with DNA methylation data is it is very highly correlated across the genome, much more so than genetic data. And so that means we're going to apply our simulation to real DNA methylation data. So we're using the Lothian birth cohort data, real DNA methylation data with the real correlations, and we've got real measures of the cell type proportions. So in our simulations, we're going to vary the number of causal probes, and we're going to vary the proportion of variance explained by those probes. So we randomly select probes to be causal, and we allocate them effect size, and we're going to allow the variance for each probe to be equal. And then for each person, you generate an error term for their phenotype, and the variance of the error terms is 1 minus the variance attributable to the probes. And then you can generate a phenotype for an individual, summing up the effects of these causal probes based on their observed DNA methylation data and adding an error term. I've got my I's and J's mixed up there. And in the simulation, one of the things when you do simulation, you can realise you can have so many combinations that you're interested in, it very quickly balloons out. So often in simulation, you'll have your baseline set of parameters and then vary one at a time for the sensitivity. So in our first simulation scenario, we took our real DNA methylation data from the Lothian birth cohorts, and we decided to allocate the causal probes just to be on the odd chromosomes. So that makes a very nice design because then we know there's nothing causal on the even chromosomes, so we can think about the null hypothesis on the even chromosomes and thinking about the alternative hypothesis when we're bringing in the data from the odd chromosomes. And because of the nature of DNA methylation data, because there's sorts of confounders, cell type proportions, environmental effects, if we simulate something that's on a probe on the odd chromosome, because of that correlation structure, we're actually going to end up with spurious associations between probes on the even chromosomes. So how do you then evaluate your method? So there's two ways that we do that. We think about the type 1 error. What's the probability that we reject the null hypothesis when the null hypothesis is true, so those false positives, and also then the false negatives. So here's one way of looking at the type 1 error. So we're looking here at this genomic inflation factor. So because we simulated our causal probes on the odd chromosomes, we can look at just the even chromosomes, and we don't expect there to be association. So we expect no inflation of the test statistics. So we expect that genomic inflation factor to be 1. So here you can see a linear model without any sorts of adjustments at all. And I think this really, if you're not used to using DNA methylation data, this really just shows you how correlated the DNA methylation is across the genome. We've put those causal probes on the odd chromosomes, and yet we're picking it up on the even chromosomes. In SNP data, genetic data, we only see this correlation between odd and even chromosomes in the context of population stratification. But this is very massive correlation structure across the genome. So some of that correlation structure is driven by the cell type proportions, and so in our analysis we can correct for cell type proportions, and you can see that that does reduce the genomic inflation somewhat, but not totally. So it's not just those cell type proportions that are generating this inflation, because we put the causal effects directly onto the probes. And you can see that the methods that we're comparing to all do a good job at controlling for the genomic inflation, but not actually as good as our method, of course. And under the null, these two new methods are actually the same. So another way that we can look at the type 1 error is to think about the false positive rate. This actually relates to a conversation we had at coffee break. That is that under the null hypothesis, we expect the distribution of p-values to be uniform. So that means that at a 5% p-value level, we expect 5% of the test to be below that level. So this then shows just how many p-values in the unadjusted case had an association p-value of less than 0.05, massively inflated. Really, these results are actually very much flipped. Qualitatively, they're the same as the ones I showed for the genomic inflation factor, but these are thinking not just about the median, but about the distribution. A last way that we can think about this type 1 error is actually now look at the results that are genome-wide significant. So by chance, we would expect there to be some genome-wide significant results, but we expect them with our Bonferroni correction to occur in only 5% of the simulation replicas. So this is then looking at the proportion of the simulation replicates that show at least one genome-wide significant association. And we were actually somewhat surprised about these results, that actually a lot of the methods are showing at least one genome-wide significant result, or a very high, relatively high proportion. But again, our method seems to be controlling that well. Okay, the next thing you do in a simulation is to think about what's the probability of rejecting the null hypothesis when the alternative hypothesis is true, or 1 minus that type 2 error, which is power. But how do you actually evaluate power in this context when you've got this massive inflation of test statistics in some of the scenarios? So the way that we got around that was to think about we used the area under the receiver operator curve statistics. So what do I mean by that? What we did was we ranked all the p-values of associations, and then essentially developed one of these receiver operator characteristic curves where if you rank the probes based on their p-value, every time we came across a true causal probe, we went up the way. Every time it's a null probe, we go along the way. So if you had very good, perfect ranking, then all your causal probes would come first, so you'd go up the way, and then your null probes would come next, so you'd go along the way. If your method wasn't doing anything and they were just randomly ranked, then you'd zigzag up this diagonal line. So anything above this, and that would be an AUC of 0.5 for random, anything above this 0.5 is showing that you have better ranking of the causal probes compared to the null probes. So the AUC statistic can just be thought of as what's the probability of ranking the causal probe higher than the null probe. And then you can see, as might be expected, without any adjustment, this ranking is not very good, but all the other methods are doing pretty much similarly. About 70 to 75% of the time, the causal probes were being ranked higher than the null probes in this simulation scenario. Okay, the next type of simulation we did was to now allocate the causal factors to cell type proportions. So because in the Lothian birth cohort data we had real cell types, we could just allocate causal effects onto the cell types, nothing to do with the DNA methylation, but because cell types are linked to DNA methylation, we'd pick that up. So it's simulating a confounding effect. And so then we'd see this spurious association between the probe and the phenotype. So this is now looking at the genomic inflation factor, and what we've got on the y-axis is the proportion of variance attributable to the cell type proportions. And so now you can see this is the analysis without any covariates, any corrections, so that's got this massively inflated genomic inflation factor. Before, when we corrected for cell types, it wasn't doing that much good, but of course we've put the causal effects onto the cell type, so that correction now does very well in this scenario. And most of the other methods, which are often very focused on correcting for cell type proportions, do well. The E-washer method seems to have an underinflation factor. That's the right word. So that was the type 1 error. And then this is now looking at this statistic where we're looking at the proportion of simulation replicates which have at least one genome-wide significant result. And now we are seeing that, again, most of the methods are performing very similarly. This E-washer method seems to have a few false positives as well as the unadjusted. But all of the methods struggle when there's too much variance associated with the cell type proportion. So if there's too much variance associated with those cell types, it's just hard to get rid of it. Okay, the third simulation scenario, we combined both of those two simulation methods where we put causal effects onto the probes and causal effects onto the cell types. And qualitatively, the results weren't different to the ones I've shown you already. But with that simulation scenario, we also thought about having a binary trait, not just a quantitative trait. So in this case, we then just bisected our phenotype, our quantitative phenotype, to make it a 0-1 trait, and basically observed the same things, that the genomic inflation factors were mostly controlled by all the methods. Ours seemed to be best controlled. And again, the ranking of the causal versus the null probes. But just to note that this is a simulation of a binary trait. In real binary traits, real case control data, you have to be really careful about confounding factors. I've been involved, I've actually generated data which has been horribly, horribly confounded, because often in disease case control situations, you might source your case samples differently from your control samples. So I did that once, not paying enough attention, and found that the control samples provided to me by a collaborator had a very different age structure than the case samples. And so I had this massive confounding, which was hard to get rid of. And in that same study, I eventually came to understand that the DNA extraction method for the cases was different to the controls. And a DNA extraction method can really impact on the bisulfite conversion step. So some words of note there, that if you are designing one of these studies, you have to be on top of the experimental design in the lab. And then this methodology, we've applied it to DNA methylation data, but it equally works on any other sorts of omics data, and this was an example of applying it, the simulations, to gene expression data. So now applying this to real data. So top left there, we've got height. Well, not surprisingly, as you saw from the previous things, there's no previous results I showed, there's no difference between the methods in height. With BMI, you now see that massive inflation, which is essentially what I presented again in that early paper from 2015. And then we can see that the correction methods really bring that inflation down. The lung function one is maybe the most interesting, and I don't know if you can see, but so all the methods, control there's no evidence of genomic inflation, but you can see some are showing massive associations, whereas our moment method, the pink one, is showing no association. So what's going on there? So here we're looking at the Manhattan plots for these different methods, and you can see that essentially all the methods are picking up very similar locations for association, but the linear mixed model methods aren't picking up anything. So what's going on? Well, this is the trait lung function, and lung function is very influenced by smoking. And so if we fit smoking status in a very simple model, then all the associations go away. So then you can ask yourself, well, is that an interesting result or not? Well, in the case of lung function, it's not particularly interesting because we know about lung function. We know that smoking is a confounder. We don't really want smoking in there. We want to get rid of it. We want to know if there's anything else associated, any other methylation associated with lung function. But you can imagine that in another context, it might be a useful result because there might be disease, if you've got a case control phenotype, there might be a confounder which is actually of interest. And then thinking about the differences between these results from this analysis versus this might actually tell you something interesting. So sometimes you have to think about what's the question I'm asking. Let's have a look at smoking. I told you about smoking. There's many DNA methylation sites associated with smoking. And you can see that all these methods, again, are picking up many. I don't know if you can see the numbers here. Well, you can. Very massive minus log 10 p-values for smoking. But you get down here to moment, and it's only actually got 3 genome-wide significant results. So what's going on? So if we take those 3 genome, you can see compared to MOA, our other mixed model method is picking up many associated sites. And so what's going on is that if we fit those 3 sites which have been identified by moment as covariates in the MOA model, all the associations with MOA go away. So essentially what moment is doing, it's purely picking up the drivers, whereas the other methods are picking up the drivers and the followers. And so again, depending what your question is, those followers may be of interest because they might be telling us about the biology, all the places in the genome that are being affected. But maybe it's the drivers that are most interesting. So it's a comparison of the methods that's useful. And then we can take these probes into prediction. And what we find in prediction of smoking status in another sample is that these 3 probes from moment actually give us the highest area under the curve in a prediction scenario compared to taking the 89 probes from MOA or the 149 probes from the linear regression. So again, that's telling us that what we're doing in the moment method is doing something useful. So now I'm going to move on to the case. How much time have I got left? Okay, great. So now I'm going to move on to my application into the case control situation. And so there's a very nice review paper talking about case control in epigenome-wide association studies, really any sort of omics studies, and again emphasising some of the issues with taking a case control design and how things can be impacted by confounders. And as I say, the other thing with the case control design is that you don't know whether you're picking up things which are causal or consequence. But in my case, I decided I was interested in applying it to motor neuron disease, ALS, which is one of the big programs I have running in my group, because the thing about motor neuron disease is that it's a late onset disease. When people turn up the neurologist, it often takes over a year to get a formal diagnosis because the diagnosis is one of exclusion. And for people, when you get this disease, it's very horrible. People often die within 2 and 3 years. It makes a difference to people if they know what their diagnosis is. And so there is a real need in that field to have an early diagnosis. And there's actually only one treatment, and not everybody goes on it. And I think if you get blood samples when people first turn up at the neurologist, when they won't have been confounded with any sort of treatment, that's a useful time to have a diagnosis. So even if you pick up things which are consequences of disease, they're not consequences of diagnosis. And the other strength of our study is that we have our own study from Australia, which has got 782 cases and 613 controls. But we were able to have a replication sample from the Netherlands. And so then you feel much more confident if you're picking up whatever you pick up in one study would be, if it's replicated in another sample, then that's useful. It's showing you something that makes sense. So in our studies that we did quite a good job on matching of age. and matching of smoking. We did not do such a good job on matching of sex, but they did in the Dutch study. So this is then the results of the comparison of the methods I've shown you. So just the basic linear regression, just to show just how much inflation there can be in these data, with the linear model with covariates, and then the MOA and moment applications. And you can see then we got several genome-wide significant results for the MOA method and not for the moment, and only one for the moment. So then we can look at, here we've got the comparison of the minus log 10 p-values for the MOA method versus the moment method, and the comparison of effect sizes. So you can see there's a very strong correlation in effect sizes. You can see that p-values tend to be smaller for the MOA method than for the moment method. We get one that survives into the moment analysis. This didn't replicate in the Dutch sample, but that might be a power issue reminiscent of the early days of genome-wide association studies. But you can also see that there's, kind of two things going on. And so, based on the simulations, my interpretation of this is this is pointing to some sort of confounder or environmental factor, which we haven't yet identified. And I guess it's then of interest is does this does this move over into the Dutch data set or not? And so we can look at that through now through prediction. So the first thing that we can look at is cell type proportions. And what you see here is, so these are now predicted cell type proportions in the Australian and the Dutch data set. Controls are in green, have green boxes, grey boxes, and cases have got orange boxes. And then we've got the Australian and Dutch. And so the first thing that we can see is actually differences in cell type proportions is being carried over from the Australian sample to the Dutch sample. We're seeing the same differences between cases and controls in cell type proportion. So in this case, often we talk about cell type proportions as being a confounder in DNA methylation analyses, but if they're actually replicating from one disease data set to another, then that's also informative and useful. So we can take these results into out-of-sample prediction, making methylation prediction scores. And what we see is that we get very significant out-of-sample prediction in all these methods. For the MOA, so when we made this methylation profile score, we did the similar sort of thing that you'd do in a polygenic risk score and take different p-value thresholds and see which p-value threshold maximise the out-of-sample prediction. Slightly winner's curse goes on there. But you can see that MOA, we had our maximum out-of-sample prediction by taking 75,000 probes. But for moment, only 25 probes gave a higher out-of-sample prediction. So to me, that's saying that the difference between MOA and moment results in our Australian study is likely reflecting some sort of technical artefact, some confound effect in our Australian samples, which isn't a reflection of what's going on actually in the disease. And you can see that the cell type proportion prediction is actually doing a little bit better, a little bit of increase when you put them together. But these AUCs are massive when you compare it to the out-of-sample prediction for ALS just based on a polygenic predictor. A polygenic predictor is based these days on a genome-wide association study discovery sample in this field of about 15,000, and the AUC from that is 0.54. And so my take-home message from this is that I actually feel like DNA methylation has been understudied as a useful biomarker for disease prediction, and one of the reasons for that is our blood samples, which have been available for genome-wide association studies across many diseases, are very they could be taken at any time point in disease. But if as we move forward and we make our go forward with better future collections, both for genetic studies and for, I think in my field of psychiatry, there's a big push for not just getting case control data, but getting new samples which have got better phenotyping, that we should also be concentrating on getting blood samples at the time that people first present, because in terms of making diagnostic predictors, that could be useful. And I feel like we should need to move away from these candidate biomarker studies, which if you ever go to clinical type of meetings, there's a lot of emphasis still on those candidate biomarkers. And what I've got here is just having a look at the risk curves for the cases and controls, and just showing the correlation between the moment and the cell type proportions, and the correlation is actually not so high, so they're picking up slightly different signals. So the conclusions of this talk was firstly to try and illustrate to a methods interested group how we use simulation, how we approach simulation, and hopefully demonstrate how it's a useful tool, and also when you read papers to really focus on those simulation results, because I think they can tell you an awful lot about the methods. I think that we've introduced these two methods more a moment, which I think are useful additions, and I think that examining the differences between the results from the two methods can be insightful. I think I've said all of that. So this talk has very much depended on the work from Jeanne. And so just to highlight that he does have this whole program suite, very much like GCTA, and the way it's presented, very nice. Many different components to it. So I've talked about the EWAS component. There's also an OREML component, which is the estimating the proportion of variance, genome-wide attributable to your omics measure. It's also got a variance QTL component, so looking for SNPs associated with variance of a trait, which is a proxy for identifying genotype by environment interaction. It's got an EQTL, MQTL component, a meta-analysis of correlations component. Often people in different omics studies are comparing correlations between gene expression, for example, and there's often when people do make those comparisons, they're not properly taking account of the errors, and this new method properly takes account of that. And so just finally, my acknowledgements. My funding comes from the Australian National Health and Medical Research Council and the Motor Neuron Disease Research Institute of Australia, as well as other funding bodies. I've depended very much on the work of Jeanne. Marta is my PhD student who's been doing the analysis with ALS. Very much linked in with Alan and Peter, Ting and Tian, Ian Deary for the Lothian birth cohorts. These are the other people in our group. These always takes $1 million to make these sorts of studies. These are the Australian collaborators for the Australian data and the Dutch collaborators for the Dutch data. Okay.